FUT2. FUT2 stands for galactoside 2-alpha-L-fucosylyl transferase 2. FUT2 creates a soluble precursor H antigen, which is an essential substrate for the final step in the soluble A and B antigen synthesis pathway. Secretor enzymes H and SE fucosylate the same acceptor substrates but exhibit different Km values. The catalytic activity for FUT is GDP beta L-fucose plus beta D galactosyl 1-3 N-acetyl beta D glucosamyl 1-3 beta D galactosyl 1-4 beta D glucosyl 1-1 ceramide. This gene is part of the protein modification, protein glycosylation pathway. The molecular functions are fucosyltransferase activity and galactoside 2-alpha-L fucosyltransferase activity. The following is a list of biological processes of FUT2. Carbohydrate metabolic process, L-fucose catabolic process, fucosylation and protein glycosylation. Other names for FUT2 are alpha-1,2-FT2 fucosyltransferase 2, GDP-L fucose beta D galactoside 2-alpha-L fucosyltransferase 2, SE2 secretor blood group alpha-2 fucosylyl transferase secretor factor. FUT2 expresses in the small intestine, mouth, colon, and lung. Common polymorphisms in FUT2 define the vitamin B12 plasma level quantitative trait locus 1, B12QTL1. Vitamin B12 is necessary for the formation of red blood cells, DNA synthesis during cell division, and maintenance of the myelin nerve sheath, among many other functions. Synthesis of soluble A, B, H, and Lewis B blood group antigens in humans is determined by the secretor SE, FUT2 blood group locus. When someone is homozygous for FUT2, RS104778 RS601338, and or RS602662, they are commonly called non-secretors of H antigen. 15% of the population are homozygous. Dr. Peter J. Diadamo states the following: as a generality, being a non-secretor does actually appear to be a potential health disadvantage. At a very basic level, being able to secrete blood type into your saliva, mucus, etc. allows for an added degree of protection against the environment, particularly with respect to microorganisms and lectins. An additional advantage of being a secretor might be a generalized tendency to promote a stabilized blood type-friendly intestinal bacterial ecosystem. Many of the friendly probiotic bacteria in your digestive system actually use your blood type as one of their preferential foods. Since secretors have a steady supply of blood type in the mucus that lines the digestive tract, their bacteria have a much more constant food supply. FUT2 non-secretors have a higher susceptibility to the following: norovirus, HIV infection, colorectal carcinomas, and Staphylococcus aureus. Being homozygous for these FUT SNPs has also been related to early tooth decay. If you are dealing with chronic illness and you are homozygous for all or any of the FUT2 mutations above, your practitioner may want to check the following: gut flora, IgG, IgA, and B12. If you have any concerns about this gene, contact a practitioner who understands epigenetics and nutrigenomics. We hope that you've enjoyed this SMP bit.